In Chapter 4, we talked about the parts of a eukaryotic cell in great detail. We kind of skipped the cell membrane at that point, and that's because Chapter 5, we're going to take a look at the cell membrane's structure and how it's used to get materials in and out of a cell. Now, the way that we currently understand cell membranes, based on a lot of work with electron microscopes and a lot of other experimentation, is a structure that we now describe as the fluid mosaic model. And that term fluid describes the fact that we do think of it as a liquid, and mosaic refers to the structure of that liquid. But it's not a liquid like water, it's actually more of an oily layer because it's made of phospholipids, as we'll talk about in a little bit. In the diagram here, we're seeing these blue structures are the phospholipids. Those are the phosphate heads that are showing here, and the tails down inside. And then the larger molecules are some proteins and some glycoproteins and some glycolipids and a few different larger molecules involved in some of the functioning of the cell membrane. So as we said, the fluid portion is made of phospholipids. Phospholipid, remember, it's a type of a glyceride, so there's a glycerol molecule that can potentially hold three tails, three fatty acid tails. In this case, there are only two tails, and where a third tail could attach, there's a phosphate group. So up here, the phosphate group and the glycerol act together as a head that we consider to be hydrophilic, meaning it will interact with water. And the tails are hydrophobic, meaning that they don't interact with water. It creates a structure that is difficult for water molecules and other polar substances to get through. And we see a double layer formed by that, where a bunch of these tails all packed together internally get a layer of the heads on both the outside of the cell and the inside. So it's a bilayer of phospholipids. And then embedded in that bilayer phospholipids are all the larger molecules, creating what looks kind of like a mosaic. If you ever made a mosaic in an art class, you can maybe kind of picture what we mean by a mosaic here. It's just referring to the overall appearance that you would have if you would be looking at the membrane. But inside that membrane, a lot of these structures would be representing proteins, sterols, including if it's an animal cell, cholesterol, and glycolipids. If you look at the word glycolipid, glyco might make you think of glucose. That's a carbohydrate structure that's attached to a lipid. So that's where that name comes from. Now, it turns out if you put a bunch of phospholipids into a watery environment, as all the tails of the phospholipids cluster together to avoid the water, you end up with a naturally occurring sphere. And here we've cut into that sphere so you can see the bilayer. If you picture kind of closing the lid here, that's the shape that naturally they would take on, just as all the tails are avoiding the water, those hydrophobic tails. And as this naturally occurs, what we have on the inside becomes the interior of the cell, and then everything, of course, on the outside would be outside of the cell. And if we think of that layer that surrounds the entire cell, the entire membrane layer, it is a bilayer, and we often refer to it as being like a sandwich, where the heads are like the bread of the sandwich and the tails are like the filling. If you were going to pass from the outside of the cell to the inside, you'd be going through first a hydrophilic layer, then a large hydrophobic layer with the two sets of tails, and then another hydrophilic layer again. Embedded in that phospholipid bilayer are a lot of proteins that aren't locked in place because this is a fluid layer. So the proteins can move around, they may slide around, they may spin in place, they may change shape in their location. Some of the proteins may be only on the outside, some may be only on the inside, or many of them pass all the way through both layers of that bilayer. So if we look at one more diagram, again showing what this might look like, we can see here some proteins that are just on one surface or on the other, or proteins that pass all the way through, such as some of these, going from one side of the cell membrane all the way to the other side. And here they're kind of peeling open which actually is how we discovered some of the things we know about that bilayer, is by actually kind of cracking open the two layers and separating them. Yeah, going back to those proteins that we find in the cell membrane. The proteins serve many different functions, and we kind of put those functions into three main categories. Those categories are recognition, reception, and transport. And we're going to talk about each one of those separately briefly here. We'll start with the recognition proteins. A recognition protein, it's all about recognizing something or identifying something. This might identify what type of cell something is, or it might help a cell be recognized as belonging inside your body or not belonging inside your body. So that might help with the immune system. And they also seem to be involved in helping cells when they kind of differentiate as your body's developing to help something like a stem cell become a certain type of tissue in your body. Now, if you remember from freshman biology, one of the terms we used often when we talked about the immune system was antigens. Well, an antigen would be a name we would apply to some of these recognition proteins, one that would tell whether or not the cell belongs in your body. The second type of proteins, we said the second job would be to be a receptor. We can call them receptors or receptor molecules or receptor proteins. And these are places in a cell membrane where another chemical can attach. Maybe it's a hormone that was traveling through the bloodstream that's going to attach to give a message to the cell. Or maybe it's a neurotransmitter, one of those chemicals that goes from a nerve cell to another cell. But this is all about communication, helping one part of the body tell a cell something specifically. The receptor site, the location we see here, is a place where a chemical can land, and that can trigger a chemical response inside the cell, because now the cell knows it's supposed to be receiving some sort of information. And the third job is transport. We further subdivide transport proteins into two subsets. There are ones like in our diagram here, which is called a transport channel protein. It's basically like a tube or like a pore, where something can go from the outside of the cell and travel through through the channel, through the pore, and end up on the inside of the cell, or vice versa, it may go outside. Also, some transport proteins are what we call transport carrier proteins. The transport carrier protein, we look at this diagram here, is something that may collect a molecule from one side of the cell. That molecule may end up inside, and the protein may actually change shape and push that molecule right through to the other side. This process may or may not require ATP. It may be an entirely passive process that happens without the input of energy, or it may require a molecule or two of ATP to kind of trigger that change in the protein to force the molecule to go through. There's kind of a short portion here with just the structure of the cell membrane and the proteins that are found in there. In part 2, we're going to talk about how things actually pass through, and we'll get into some detail of things like diffusion, osmosis, and active transport. As always, if you have questions, please bring those questions to class, and I strongly encourage you to read through Chapter 5 in your book, or at the very least, look through and look for ideas that you're struggling with or things that were not part of this video. And have a good day.